So this is the third part of lecture 1, and in this part I'm going to talk about types of enzyme specificity. So anyway, you already know why enzymes are so special, because by using enzymes we can achieve higher reaction rates, because as enzymes increase the reaction rates tremendously. Okay, and then we can make the reactions to occur at milder reaction condition. For example, at lower temperature. I mean, say sometimes say, for example, one reaction it requires high temperature, but using enzyme we can lower this temperature. I mean, at low temperature the reaction can occur in presence of enzyme. And then enzymes are very specific for the substrate and products. That means enzyme binds with a specific substrate and produce specific products. And finally, capacity of control. We can control the speed of the enzymatic reactions. So okay, anyway, so from where this specificity comes? I mean, why enzymes are very specific? Okay, there are actually two proposed model. One is lock and key model, and another is induced freeze model. First, the lock and key model. The idea of this lock and key model came from a scientist named Emil Fischers. Okay, so in 1894, Emil Fischers made a discovery, and he observed that glycolytic enzymes can distinguish between stereoisomeric sugars, led to the formation of his lock and key hypothesis. Okay, so and the hypothesis is the specificity of an enzyme, that is the lock for its substrate, the key, arises from the geometrically complementary shapes. Okay, so if you see this figure, so say this is the enzyme and these are the substrate. So this substrate bind here, this part of the enzyme. So if you can if you look at the shape, they are complementary to each other, right? Like this one will bind here and this portion will bind here. Okay, so this is lock and key model, that the shape of the substrate is complementary with the shape of the enzyme binding site. Enzyme binding is in the substrate binding site of the enzyme. Okay, substrate binding site is that part of the enzyme where the substrate bind. Okay, so based on according to this lock and key model, the substrate binds with the shape complementarity site of the enzyme, and then catalysis occurs. Okay, so the site where the substrate binds in the enzyme is also called active site of the enzyme. Okay, so anyway, so usually the non-covalent forces through which substrates and other molecules bind to enzymes are similar in character to the forces that dictate the conformation of the proteins themselves. Okay, and the non-covalent forces which are involved are van der Waals, electrostatic, hydrogen bonding, and hydrophobic interactions. Okay, so these non-covalent forces are involved in the substrate enzyme binding. Okay, and a substrate binding site in the enzyme consists of an uh, indentation or cleft on the surface of an enzyme molecule. Okay, so if you look back to the figure, so this is the substrate binding site. It's an indent, right, or cleft. Okay, and this substrate binding site, okay, they have some characteristics. So the number one, those sites are complementary in shape to the substrate. This is called geometric complementarity, and also their shape, that side can have electronic complementarity. Okay, so that means that there will be complementarity in regards to the charge. Okay, so like how this is achieved, the amino acid residues that form the binding site are arranged to interact specifically with the substrate in an attractive manner, that is electronic complementarity. Okay, so the substrate binding site of the enzyme usually they show this two type can show this two type of complementarity: geometric complementarity or electronic complementarity. Okay, now the induced fit model. Okay, So according to these models, the enzyme active site, where the substrate bind, is not very rigid, is not completely rigid fit for the substrate. In the lock and key model, the those sites are exact complement. Okay, but according to this induced fit model, those are not exactly complementary. Rather, the active site of the substrate binding site of the enzyme, they changed according to the shape of the enzyme, shape of the substrate. Okay, and this model actually it has two advantages if we compare with the lock and key model. So what are those advantages? One, this model explains how enzymes may exhibit broad specificity. Okay. So if an enzyme's substrate binding site is very rigid, it can only bind with the substrate which has that exact shape, right? But if the substrate binding site in the enzyme is not rigid, that means it can change according to the shape of the substrate. That means it can bind to many different substrates. Okay, so if it is possible, then that enzyme can show broad specificity, right? Okay, and another advantage is this model explains how catalysis may occur. Conformational changes stresses bonds in the substrates, increasing reactivity. When the substrate binding sites changes its shape, which is called conformational change, the bonds, okay, the chemical bonds, they also get some changes or stresses, which ultimately can increase the reactivity, the reaction rate. Okay. Okay, so this was two model, induced fit model and lock and key model. And in terms of specificity, enzymes show four distinct types of specificity towards their substrate: stereochemical specificity, absolute specificity, group specificity, linkage specificity. Okay, in case of stereochemical specificity, the enzyme will act on a particular steric or optical isomer. Okay, in absolute specificity, the enzyme will catalyze only one reaction. In group specificity, the enzyme will act only on molecules that have specific functional groups, group specific, okay, such as amino, phosphate, or methyl groups. Finally, in case of linkage specificity, this enzyme will act on a particular type of chemical bond, particular type of chemical link, okay, regardless of the rest of the molecular structure. Okay. In the next few slides, I'll talk about the examples of this specificity. Maybe it will be more clear then. Okay. So then first, the stereo specificity. Okay. And in these cases, the enzymes, who shows stereo specificity are highly specific, both in binding chiral substrates and in catalyzing their reactions. Okay. And enzymes are absolutely stereospecific in the reactions they catalyze. Very specific. Okay. So here is the example. For example, the L-alanine oxidase. This is the enzyme L-alanine oxidase. It's only react with the L-alanine, not with the D-alanine. Okay. L-alanine, D-alanine, both are alanine. But this enzyme L-alanine oxidase, it only recognizes the L-alanine. Okay, so this is stereospecificity. This enzyme only recognizes this steric structure of alanine, the L structure. Then geometric specificity. Basically, geometric specificity also belongs to the stereospecificity groups. Okay, so in case of geometric specificity, a substrate of the wrong chirality will never fit into an enzymatic binding site. Okay, and most enzymes are quite selective about the identities of the chemical groups on their substrates. That means these enzymes, who shows a geometric specificity, they require a specific chirality. That's a specific structure, and in addition, they also require some certain chemical groups. Okay, so this make this geometric specificity more stringent. Okay, so that means geometric specificity is a more stringent requirement than is stereospecificity. Because in case of stereospecificity, those enzymes only need particular stereo-specific structure, particular chirality. But in case of geometric specificity, they require both particular chiral structure, also particular groups. Okay. Okay, and so enzymes are considerably in their degree of geometric specificity. One example is YADS. YADH. It's a enzyme and it catalyzes the oxidation of small primary and secondary alcohols and change them and produce aldehydes or ketones. Okay, but this enzyme, they are most efficient for ethanol. Okay, so when they catalyzes ethanol, it's very fast. But when they catalyze methanol or isopropanol, those rate of those catalytic reactions are 25-fold or 2.54 slower than the rate of ethanol. Okay. So this figure shows they depicts that example which I have just talked about. So ethanol enzyme reaction will be very fast. Methanol enzyme reaction will be slower. Okay, then bond specificity of enzyme. So for example, a peptidase. Okay, so peptidase enzyme, they are specific to peptide bond which are found between amino acids. Okay, so peptidase, they are not specific for the amino acids. Okay, so they don't care which amino acids are forming the peptide bond. Okay, they only care about the peptide bond. If there is peptide bond, peptidase enzyme can recognize that bond and break it. Okay, such as for example, this peptide bond, okay, formed between glycine and glycine. This peptide bond formed between valine and alanine. Peptidase enzyme will, break this peptide bond in a similar speed. Okay. Finally, group specificity. Okay, so in this type of specificity, the enzyme is specific not only to the type of bond but also to the structure surrounding it. Okay, please. Okay, so please try to understand. In case of bond specificity, those enzymes, they only care about the bond. They don't care which are around. Okay, but in case of group specificity, they recognize uh, the specific bond. Also, they basically recognize the surrounding structure. That means the surrounding structure has effect on their activity. One example is pepsin. Pepsin is an endopeptidase enzyme. Endopeptidase enzyme. Okay, so it breaks the protein from inside. Not from the outside. Okay, so pepsin is an endopeptidase enzyme, and that hydrolyzes central peptide bonds in which the amino group belongs to aromatic amino acids. For example, phenylalanine, tyrosine, and tryptophan. Pepsin, they also act like peptides. They can also break bonds, the peptide bonds. But for them, only presence of only peptide bonds is not enough. Okay, that means those peptide bonds should be formed. Belong to the amino groups. Okay, to aromatic amino acids. Okay, so that is all for lecture 1. Okay, see you in the lecture 2. Till then, be safe. Thank you.